This is an additional video for the students doing higher GCSE science. Make sure you've watched the previous two videos because I'm just going to give you some additional information here. So the first thing we're going to look at is some extra information about meiosis. So we understand that meiosis has two splits and it makes 4 gametes. But how are all them gametes genetically different? So each sperm or egg cell has completely different alleles in to give variation. So we are completely different from brothers and sisters. So how does this happen? So I'll show this again with 4 chromosomes. These are in 2 pairs. So we'll say that the green and orange chromosome there in pair 1 and the red and blue there in pair 2. So a sperm or an egg cell only needs to get one of these pairs. So it needs either a green or an orange. So when these split, one of the cells could get a green from pair 1 and a red from pair 2, but it could also get a green from pair 1 and a blue from pair 2. But because we've got 23 pairs of chromosomes, there's lots and lots of different combinations that can happen here. And another thing which leads to even more variation is, so in that pair 1, you got one of them chromosomes from your father and one from your mother, but they can actually swap parts of the chromosome between each other. So here a little bit of the chromosomes got kind of mixed up. So you've got one chromosome that's mainly from your father, but it's got some of your mother's alleles on. And this can happen with all of the chromosomes. So that when the first split happens, you get a mix-up of the genes. And then when the second split happens, you get 4 gametes that are completely different from each other. So this can be incredibly complicated. So no 2 gametes that have been made would ever be the same because there's so many different combinations that can happen. So meiosis leads to gametes that are genetically unique. They've got completely different DNA from the parent cell, and this leads to the enormous amount of variation that we see in humans. We're all different from each other because of this process. And this is completely different to mitosis, which makes cells that are all exactly the same. So when we were made in the zygote and we grew, all of our cells have exactly the same DNA in. That's because mitosis creates cells that are genetically identical. Okay, so in the previous videos we looked at recessive diseases such as cystic fibrosis. And in cystic fibrosis, if you had one copy of the diseased allele, then you didn't have the disease, you were a carrier. And that's because the dominant allele would overpower that. And the dominant allele in a recessive disease is the healthy one. So what would happen if the dominant allele in a disease was actually the diseased one? And this is the case in Huntington's disease. So this is caused, we'll call that allele H. And if you have one copy of the big dominant allele, this is actually the diseased one. So you would have the disease even if you've just got one copy of this diseased allele. So a question you could get on this would be, how could 2 people with this disease have a child that did not have it? And that is a very similar question to the one where 2 people that didn't have cystic fibrosis can have a child with cystic fibrosis. So 2 people that had the disease would have a 25% chance of having a child without the disease. If only one parent had the disease, then there would be a 50% chance of their child having the disease also. And the final thing we're going to look at is the Human Genome Project. So the word genome just means all of the genes. So this was a project that found all of the genes in humans, and this was completed in 2003. So they mapped out all of the A's, C's, T's and G's that are found in humans, and in each of our cells we have 3 billion of these bases, and these are found on 46 chromosomes or 23 pairs of chromosomes. So this is what our chromosomes look like, and you'll notice that the 23rd pair, that can either be XY or XX, and these are called the sex chromosomes because they determine your gender at birth. So what are the uses of knowing all of the genes that make up a human? Well, the first one is we can figure out which genes cause specific diseases. So we know which genes cause cystic fibrosis and Huntington's disease, but we can also find out genes that cause certain cancers or heart disease, genes that make it more likely for people to get Alzheimer's disease and to have high blood pressure, and so on. And the second use, so this can then be used to spot diseases before they happen. So Angelina Jolie did a genetic test and she was found to have a certain gene, which is called the BRCA1 gene, and this means that it was 87% likely for her to develop breast cancer in the future. So she made the very brave decision to have her breasts removed so she could then not develop breast cancer, and this has led to many other diseases where you can spot a gene and you know that you are more likely to get that certain disease. You can put things in place to make sure that you do not get that disease in the future. So the first two uses, figuring out which genes can cause diseases, but then it can also be used to help cure and treat genetic diseases also. And the third use is this idea of personalised medicine. So at the minute, if you have a certain disease, so the example here is colon cancer, everybody at the minute gets the same treatment for that type of cancer, and on some people that treatment works, on other people it doesn't have an effect, and on some people it actually makes them worse. But in the near future, what they think might happen is they would take a look at the patient's genes and also which specific mutation they have that caused the cancer, and they'll know if you had these sort of genes, this is the best treatment for you. People with these sort of genes, well, if you have that same therapy, that's actually going to cause you harm, so try this therapy instead. And this is the idea behind personalised medicine. So everybody gets a treatment which has the best effect for them depending on their genes. And this has led to further studies. So in 2003, the first person had their entire genome sequenced. In 2012, 1,000 people had their genome sequenced from around the world. And in the UK in 2018, they sequenced 10,000 people's genomes. And the more people whose genomes you sequence, you can find out more rare diseases and unusual causes of certain diseases. And this advance is actually because computers are getting better. So it would take quite a lot of computer power to compare 3 billion letters in 10,000 different people. But this can be used to figure out rarer and rarer diseases. It can be then used to look for treatments for these certain diseases. And it also means that personalised medicine can be the treatment in the future that will have a huge amount of benefit for every patient. So if you are a higher student, please try some questions now. If you're doing the triple course, there is additional information that you need to know, so watch the additional video. Thank you very much for watching, and good luck for your up and coming test. See ya.